Nuevamente les doy la bienvenida a este nuevo webinar que hemos organizado en conjunto con la Comisión Directiva de la Sociedad Argentina de Inmunología y principalmente con la Comisión de Docencia, que realmente está teniendo un rol muy activo. Probablemente en el momento que ustedes hayan ingresado a nuestro canal YouTube hayan visto también un flyer recordándoles el concurso de mini videos que hemos organizado, que la Comisión de Docencia también ha organizado en conjunto con la Academia Nacional de Ciencias para celebrar el Día de la Inmunología. Es un concurso de mini videos en donde pueden participar los estudiantes de todos los niveles de escuelas públicas y privadas de la República Argentina. Así que bueno, les pido por favor que difundan la actividad, que creo que puede ser muy rica, muy nutritiva y puede servir mucho para difundir la importancia de la inmunología en estos días y en todos los tiempos. Bueno, la actividad de hoy nuevamente está enfocada en aprender y compartir experiencias sobre la enseñanza de la inmunología, y hoy tenemos a Mallary Greenlee-Wacker, que va a ser introducida por los doctores Cecilia Rodríguez Galán y por el doctor Antonio Carrera Silva. Así que bueno, los dejo con ellos y les agradezco muchísimo su participación. Espero que esta sea una jornada sumamente enriquecedora. Los aliento a que hagan preguntas y a que conversemos, y así puede ser lo más relajada, distendida y fructífera posible. Bueno, muchas gracias.
Bueno, muchas gracias, Mariana. It's a great pleasure to me to introduce Dr. Mallary Greenlee-Wacker. She's an assistant professor at the Department of Biology in the College of Science and Engineering at Central Michigan University. Dr. Walker is a professor of biology, and her expertise is focused on the immunology field, particularly in the innate immune response, inflammation, and the host pathogen interaction. The main goal of her research laboratory is to understand how the bacterium Staphylococcus aureus perturb the ability of human phagocytes to effectively kill ingested bacteria and regulate excessive inflammation. She has published several papers in prestigious journals as Blood, Journal of Immunology, Journal of Leukocyte Biology, among others. Dr. Walker is genuinely passionate about undergraduate education and research. She teaches courses in cell biology and immunology, and she uses active learning strategy to increase understanding, engagement, and retention. Dr. Walker has mentored over 20 undergraduates today. She has also developed a research symposium for undergraduates at CMU, and she's the undergraduate research coordinator for the Midwest Autumn Immunology Conference, and ultimately, as a mentor and instructor, she hopes to cultivate a long-lasting interest in learning science and immunology. Welcome, Mallary, and thank you for accepting this invitation, and we are in your hands now. Thank you very much.
Well, thank you for the invitation to talk about what we do in the classroom. So I'm excited to tell you a little bit about the games we play. Let me share my screen. All right, can you see my screen okay?
Yes, we are seeing.
All right. So I'm at Central Michigan University, which is in Michigan. So it's the state that looks like the shape of a hand, and we're right in the center of that hand. And so I came to CMU 5 years ago. And at that time, the biology department had just redone their curriculum, and a big emphasis was placed on student-centered learning. And that was because of the Vision and Change initiative that came out of the NSF. And so we wanted the student-centered learning, and a lot of faculty had committed to teaching in this active learning format. So doing activities to reinforce difficult concepts. I also like to use active learning because of the literature. And so Scott Freeman's written a lot of really nice papers on the benefits of active learning, just to highlight 2 here. So active learning has been shown to increase examination performances and decrease failure rates by 55%. It's a huge number. And because it also is disproportionately beneficial for underrepresented minorities from low income backgrounds. So it's a good thing to use. It's also really fun. And so I really enjoy teaching this way and hearing the students get really enthusiastic about the content that I'm teaching. So when I teach active learning, I'll explain it to my students and I'll show them these pictures. And so you have the Peanuts characters, you know, absorbing information from their teacher, right? With the wah, wah, wah. Or if you've seen Harry Potter, Snape is your classic traditional lecturer. And then you have an active learning format that's a lot more engaging and students are really getting to work with that material. So a little bit about my immunology course. So my major goal for the immunology course is to emphasize critical thinking rather than memorization. So I really want them to get at the heart of what we're talking about. And so to do this, I want to make the material easier to understand and easier to recall so that it's easier to synthesize. So one of the problems with immunology for students is that it almost seems like a foreign language with all the CDs and the ILs. And so I want to make that material easier to recall and understand. My class is around 18 to 24 students. It's an upper level class. And because of the commitment to active learning at CMU, almost every one of them, by the time they end up in immunology, has taken an active learning class here before. The majority of my students identify as pre-professional, so they want to be doctors or pharmacists, dental students, pre-dental students, but less than 10% would like to pursue a graduate degree. Just to note, I have used a very similar active learning class when I teach my introductory cell biology class, and that has around 70 students in it. So the activities I'm going to talk about, some of them are scalable for a very large group. So my class is taught in a blended style. And what this means is that I'm going to do a little bit of lecturing along with some activities. So before class, I expect that students either read the textbook or watch a video lecture, and I give them a note-taking guide to take down the important notes from either the text or my lecture. Then when they come to class, they're expected to take a quiz over that material. So they have to come prepared because they're going to take a quiz. And I've used a couple different formats for the quiz. So during the pandemic, when we were virtual, I used our Blackboard interface, which is just our online interface, and there was a quiz that students could take at the beginning of the class period. But what I really like are these scratcher cards. They're IFAT scratcher cards, and you can code your questions so that the right answer has a little star under it. And this is nice because the students can get feedback right away that they've gotten the wrong answer and they can scratch again. And so I might count like the first guess as 50% off, but if they get it on the second answer, then they might just get 0.5 points, which is a little bit lower stakes for the students. And then, so that takes around 10 minutes because it's around 5 questions. And then I will go into a mini lecture, and I spend around 15 minutes doing my mini lecture. So if this is the class about VDJ recombination, that means that I'm going to hit on the important points of VDJ recombination, just bring it back one more time for the students. Then we'll do our activity. The activity I'm going to show today takes around 20 minutes. It's a modeling activity. Sorry, my lights go off. And then right after the modeling activity, I'm going to do a short assessment question. So this will just be a little polling question. So I can poll if we used Microsoft Teams, but you can do this obviously in Zoom. In class, we've used the eye clickers before if we're in person. Then I do another activity. That's a mini presentation activity where the students all break up into groups and do these mini presentations. And then I have 5 minutes to wrap it up. So that's the gist of the lecture. So this was one of the first activities I really decided to tackle in a serious way, VDJ recombination. And I really wanted to think of an activity that would model this process. And that's because this topic is really challenging, and it reappears multiple times through a basic immunology course. So you see it here, you see it again during T cell and B cell development, you see it during affinity maturation. And so I really wanted them to get a concrete idea of what was going on. It's also shown in textbooks as a very static process. And so you might see a figure like the one I have here where you just have all these things going on, and it doesn't really show the process occurring in time. So my objective for the students is to explain how the following events contribute to antigen receptor splicing and diversity. And I want them to be able to explain how gene segments, how the process of recombination, and how light and heavy chain pairing contribute to this massive amount of diversity that we get. So the materials that I use to do this activity are Legos. And so you can get like a Lego Classic Basic box, or even a medium creative brick box. Those are the names of just some like random Lego sets. I had these laying around my house on the floor, so I just scoop them up. You'll also want some labeled enzymes. I'll go over these in a little more detail. Labeled Ziploc bags, and I have these labeled by their gene segments, and then a Lego base plate. And if you want to screen capture this link, there's a PDF of this activity on the SLB Learning Management site, and it should be available to everyone. If you have difficulty, you can always reach out to me, and I'm happy to send out that PDF. So here are the materials just shown in a little more detail. So I have bags filled with Lego bricks that represent the V, D, and J gene segments. And so they're labeled as V, D, and J. And then I have flanking those letters, those gene segments, I have the recombination signal sequences. So you have your heptamer, your 23 base pair spacer, and your nonomer. And I emphasize to students that this is where the RAG enzymes are going to bind, right? And then we have all these different blocks that represent gene segment, and at random, one of those is going to get pulled out. I also have a bag of nucleotides, and so you'll want 4 to 5 colors. These are the single pegs, 1 color for each A, T, C, G. And then if you want to use this again for affinity maturation, you want 1 more for uracil. Then the enzymes, and you can add enzymes if you want to focus on more, right? Obviously there's more enzymes to this process. It depends how specific you want to get in your course. For me, this is kind of the enzymes I want them to know the function of. So I have an exonuclease, Artemis, RAG1 and 2, DNA ligase, TDT, and a DNA repair enzyme. I just turn a block and label the other side. So it's not shown on this picture here. And here I'm using Duplo Legos, but you could really use anything for these enzymes. I don't click them onto the base plate. So the first step I show is, and I want to emphasize to the students, is that RAG1 and 2 bind to the recombination signal sequence. And this promotes the hairpin formation. And pre-COVID, I would have a student come up and at random grab a D gene segment and a J segment, and I would emphasize this process is totally random. And then I tell them that if this is a light chain, we'd begin with different gene segments first because the order is slightly different in those 2 chains. So then I'll show them that that RAG binds, right? You have these random gene segments, and it promotes, right, the RAG 1 and 2 clips, those sequences, and it promotes the formation of a hairpin. And then I make a hairpin with my pegs. And so here, my little hairpin, right, which is the white and green, and then the white and green in the opposite way. So RAG 1 and 2 nicked, right? And I'll show them this. And then we have the hydroxyl attaching to the phosphate here, and this creates this little hairpin. So you kind of have to imagine that this is closed. Then I'll tell them, I'll grab the Artemis enzyme, and I'll tell them that this will eventually create P nucleotides. And so I'll bring this enzyme in, and I'll show them that this often cuts unevenly. And when it cuts unevenly, so if it were to cut here, then the sequences that were on the bottom get flipped to the top. And so we had white, green, and now this white's going to go here, and this green is going to go there. And I'll show this to the students by adding those pieces to the top. And then I'll tell them that the DNA repair enzyme then comes in and adds complementary sequences. And because of the nature that Artemis opens this hairpin, we're left with palindromic nucleotides. So sequences that are read the same forward as backwards. So if we read the sequence as green, white, then they're read backwards as green, white, white. And then I'll point out that this is new sequence. This is new sequence that's been added to this signal, this junction here, right? So we've added diversity. So we had our gene segments, and now we've added more diversity by this process I just described. And then I'll explain to them that this is also, right, I usually only show this on one gene segment, but this is also happening on the opposing gene segment. And then those are joined together by DNA ligase. So if this is a light chain, this is V and J being joined together. And now you have all this extra diversity within that segment. In the heavy chain, I'll again explain that if we start, there's a little more complexity here. So here we have Artemis opening up the hairpin. And before we have those P nucleotides being filled in, exonuclease comes and removes at random, right, some of these nucleotides. And then an enzyme called TDT will at random add more nucleotides. So this is creating new sequence, and it fills in all those nucleotides. So here we have now P nucleotides, but we're missing some. And then we have our non-templated nucleotides. So this was sequence that was never there in our genome. So these are the N nucleotides. And so this is a very clear picture of new sequences or new sequence information being added to that junction. And then I'll come and add them. The ligase will come in and join these sequences together. And then I'll say this happens. So here we have D and J, and this happens again with the V gene segments. And we have the whole process starting over again. And then I'll point out to the students that this is our DNA sequence. So a lot of DNA has been lost. And now there's this new DNA in each one of these clones of our lymphocytes. And this is going to get made into RNA, which is going to get made into proteins. And that all of the sequence will encode for proteins that are found within the variable region of our antibody or within the variable region of our T cell receptors. So this is where that antigen specificity comes from on a DNA level. So that's the end of that activity. We then, as I said, have a quick polling question. So I might ask the students in a polling question, what would be the expected results of RAG deficiency, right? So they should be able to explain that VGJ recombination wouldn't occur, or we wouldn't have antigen receptors. And then I have the students do these antibody mini presentations. So each group is assigned one topic that is a way that the immune system increases antibody diversity or antigen receptor diversity. And so a group will get the action of Artemis or gene segments, or the action of endonuclease, or the action of TDT, And then they have to come up with a creative way to present the material. So it could be role play, it could be drawing out an animation virtually, and then they film it. And then I compile all the presentations under the class resources. And I'm not sure if this audio is going to work, but I don't.
Heavy and light, heavy and light. I gotta have me my heavy and light. The mu, the delta, the kappa.
I'm not sure if that came through, but one year I had 2 students, Justin and Justin, make a rap about the heavy and light chain, and it was quite comical. And of course, everyone remembered that on the test. So we always have weekly homework. And so for the homework, I asked them to do a multi-step process that walks through recombination. And then for the exam, I give them just an open-ended exam question. So I ask them to basically describe the entire process of VDJ recombination and explain at what steps receptor diversity increases. And so they usually do pretty well on this class, and they're able to recall this test, and they're able to recall the information. Just a comment real fast. I also bring this up again for affinity maturation. So I have my AID enzyme, and so I'll show them that AID can replace a uracil here, and that DNA repair enzymes can introduce new nucleotides during this process. And then I have my little constant region here, and I'll explain with the AID enzyme how isotype switching works during affinity maturation. Just a few other activities that I do in my class. So we do a lot of traditional active learning things like think, pair, share, jigsaw, case studies. I do Jeopardy and Taboo for helper T cells. Sometimes I'll put an animation of an immunological process on mute, and I'll have them describe the process. And so this is something that I learned about in an active learning workshop from a foreign language professor, and I thought it was really cool. I also do these what's wrong with this picture from the Children's Highlights magazine. And so I'll have the students diagram a process, and then they swap with each other, and then they find the mistakes. And then on the exam, I'll show them a process where there's mistakes in it. And so this is antigen presentation, but I have HLA DO labeled as the invariant chain. And so they can find the mistakes and solve these puzzles. I like to do a lot of these interrupting the pathway exercises where I'm with signaling, for example, I'll tell them like I added an inhibitor, what goes up and down. Or in complement, right? I deleted the C1 inhibitor, what goes up or down. And so I'll even give them the pathways. And this way, they, instead of just having to memorize those pathways, they have to actually think about how everything's connected. Sometimes we do a little bit of storytelling. So I might have them give them a little story about the immune cell detective. And there's 2 detectives, Mac and Mr. T, and they have to solve a crime, and the perpetrator gave them terrible food poisoning. What clues were left? And so I tell them this, and they have to look at, this is a picture of salmonella, and so they have to look at what TLR clues were left behind that told the immune cells what was what it was. And then finally, we do Pokemon battles. So I'll have them. I'll show them a Pokemon card, and I'll explain that, you know, on every Pokemon card, there's some information about the type of stage it's in. So differentiation, essentially. There's a picture and there's a tax, so much like an immune cell. And so I'll ask them to make these for immune cells. And then I'll ask them to tell me how much each immune cell activity takes in terms of time. So if it's an innate, it doesn't take a lot of time, but if it's adaptive, it's going to take time to act. And then I play pathogen cards, and I try to get the students to play, when I play a virus, to get them to play helper T cells or cytotoxic cells or NK cells. And then when I play a bacteria, they should play a phagocyte or a helper cell. And so I try to get at that idea of humoral immunity and tailored immune responses through this activity. Just a few acknowledgements to end. So I've gotten a lot of guidance from people at CMU who are very good at this style of teaching, in particular, Dr. Steve Gorsuch and our Office of Curriculum and Instructional Support, and my kids for giving me a lot of these ideas.
Thank you very much, Mallary. It was very nice talk, really. I enjoyed it a lot. I think we have a lot to learn about the new strategy to teach immunology. I think it is very, very interesting. We have one first question that is related to the how many classes per week do you have, and how long is the course that to teach in this way, trying to apply this concept?
Yes, so I have, I meet twice a week for 90 minutes each meeting period. So sometimes to cover everything in immunology, sometimes I will lecture more and have activities less, or the activities will be much shorter so that I can cover some more content. So for example, if we're covering immunodeficiencies, I don't have as much time, right, to get through, or if we're covering allergies, right, and I want to get through the different types of, or hypersensitivity, and I want to get through the different types, I don't have a lot of activities for those days. So I kind of ebb and flow, and the students seem to be flexible. And then the courses are around 15 to 16 weeks.
And how was the response of the students when you come with this new strategy? What was the response to this new strategy, or to this new idea of learning in this way?
So I'm lucky in immunology because, like I mentioned, a lot of the students have had active learning at CMU before. And so, but what I always do is I give them the data. So I give them the data that shows how active learning increases or decreases failure rates. And I explain to them also that by observing them problem solving and interacting in this way and interacting with groups, I'm able to write better letters of recommendation because I can actually see them problem solve and synthesize information. And so knowing that, they're pretty receptive to learning in this way, and they like to take a lot of those breaks. Sometimes with the role playing, I'll get students that don't really want to engage in that way, and I don't force it. You know, I always try to make something available, especially virtually. You know, you can log in the chat and put your comments in, or you can like it, so that I don't put them on the spot if that's not their preferred way of learning.
What was the adherence of the students in percentage, more or less?
Sorry.
The adherence. How many? The percentage of students that like it or doesn't like it? Yeah.
I think most of the feedback I get is positive. So. You know, 90% of the students like the way the course is organized. You know, so they're just used to not getting the lecture-based. Well, I think it's because I also will include video lectures, so that's a big component. So they're able to get some of those on those mini lectures I put in my courses. So they're still able to get some of that traditional lecture material. They don't have to be on the entire class period. That would be really, I think, tiring.
Okay, I have a question for you, Mallary.
Yes.
Can you hear me?
Mm-hmm.
Yes. This is a really challenge for most of the professors that usually we don't use this strategy, and this is really novel for us. So I teach mostly T-cell ontogeny. So while you were talking, I was thinking about how creative I had to be to to develop some of these or adapt some of this teaching to this new strategy. So there is, I have a million questions. For example, how do I start to incorporate this method? There is bibliography available. Is there any subject in immunology that you can adapt to this active learning? I mean, I don't know how to start.
Yeah. So when I started teaching immunology, this is where I started, right? So I started with one activity that I thought was, as an immunology student, tough to get. And so I made that activity. And then I flipped a course from the start, which was a lot. And certain things are very adaptable. So things like the what's wrong with this picture and the interrupt the pathway, you could take anything and do that, and the students can work on it and solve that. So not every activity is going to work.
Right.
But I say even if you start with one or two in a semester, if it's a tough thing, the student, especially modeling, the students will benefit from it.
Right, and there is some bibliography. I mean, there's other people that publish this active learning. I mean, you base all your knowledge or you develop your own creative activity, or you just also have some other professor that develop also and incorporate that to their courses.
Yeah, I was lucky. I was lucky or naive. And there's a lot of literature. But I was lucky in that people around me were doing a lot of this and had been for a long time and knew the literature, and I could just understand things from them. So like the write your own pathway, find the mistake. Someone was doing it with 2 DNA molecules where they would put mistakes in how they align, which was cool because then you explain that's how AZT works, right? So you can't then add. So it became very interesting to the students. And I thought, oh gosh, like when you can then work drugs into it, right? And so I had people doing it around me. So not to say that, there's not a ton of resources available.
Yes.
Yeah, we have some comments on the chat that I will read to you. There are people that saying that our experience at first year of college, when they are just finishing high school, they feel too cold to do this type of game, and of course they laugh. They don't. Sometimes they reject to do this. But we need to find the way to motivate, or they find that they accept, or they actually like it, that it is a good thing.
So one of the things I do too, and I forgot to mention this, when I do it's always good if you can do it beforehand, right? If you can have your data for your class beforehand, if you've been teaching a class, then introduce the activity and show them the learning gain from that. That's one way that you can create buy-in for doing these, for convincing them that this is the cool thing to do. But one of the things I always do is I have a clicker question right after that they're able to synthesize. And then I show them like, gosh, you guys all got this, right? And even if they don't, it's fine because I know where that immediate assessment, right? But if I point out their learning gains to them, they're pretty happy.
Very receptive. Yeah. And there is one more comment that someone said that I use a variation of what is wrong with this graph and what is missing from this graph, and it said that worked very, very well and great.
Yeah, I saw someone asked if we have lab activities. We don't teach an immunology lab. We teach a cell culture lab where it's like inquiry-based type stuff and independent projects. It's a more typical lab experience.
Mm-hmm. Yeah, I think Ceci can comment this because in immunology here we have a really active laboratory working things for students. I think it's, and that is the part that they really like it in the laboratory part of it.
Exactly. I think it's more adaptable to our lab activities. I mean, because there's smaller group. We have these big lectures. I mean, our university is free, so there's a lot of people coming to university. So sometimes our lecture classes are 80, 100 students. So that might be very complex to adapt, but the lab classes are a smaller group with younger teachers that I think they really will like to adjust this kind of learning. Very interesting. I think you have to be also very creative.
Yes, I think we have to put a lot of imagination in
Right.
and creative. But I like it. I think it's a good way to really to teach.
So how do you measure? Sorry, I mean, I don't know. We have time for questions, but how do you measure? I mean, you said at the beginning that this is 55% better acquisition of knowledge that the other. How do you measure that?
So that was a study by Scott Freeman. And what he did, the way he designed his course is that he had this very large course. And he implemented active learning in half of it and didn't change the other half. And then he compared the failure rates in a high failure class. And so those numbers are from.
Very interesting.
Yeah.
Mariana, I don't know if we have time for one more question or we need to continue for the next speaker.
There's one more question. I'm trying to. There you go. I don't know if we have time. Nicholas is already here, but maybe this is the last question. And thanks so much, Mallary, because it has been really very challenging for us, very creative, and so it's great.
Yeah, okay, I will read the last question. Did you receive feedback from the teacher of superior course? They feel their student more eager or better from format after having active learning in immunology?
I get lots, not an instructor, but I get lots of feedback from past students saying, you know, in medical school, in dental school, in PA school, saying, oh my gosh, your class prepared me so well. Everyone's struggling so badly, you know, but I don't know. I mean, that makes me feel good, but I don't know if it's because those students took immunology or they didn't, you know, but they say they can recall the stuff still. So that's good news.
That's great. Yes, of course. That is the purpose of teaching. That lasts forever.
Yes.
Mallary, that was great, your class. I really enjoyed it.
Thank you.
Thank you, Mallary, very much. And we have our next speaker, Dr. Nicholas Pullen. Nicholas, thank you so much for being here with us today. Cecilia Rodriguez and Antonio Carrera Silva will chair the session. And again, thanks for being with us today. We wish we could meet you personally here in Argentina, but well, here are the circumstances. So we will try to share experiences with you and to learn how you motivate your students. Thanks.
Alrighty, so you should be able to see my screen now.
I was ready to.
Okay.
Nicholas, one
Yeah.
minute, please. I think Cecilia is going to
Wait.
introduce.
Okay,
Wait a
sorry.
minute. Yes, can you just get out? I can't see my own computer. Okay, there you go. Sorry, Nicholas. I wanted to introduce you first before your talk. I have the pleasure to introduce Dr. Nicholas Pullen. Dr. Pullen is currently a tenured associate professor at the School of Biology Sciences at the University of Northern Colorado. During his time as undergraduate, doctoral, and postdoctoral student, he has acquired great experience in the field of biology, anatomy, and neurobiology, and more recently on molecular immunology. His research work focuses mainly on the interdisciplinary applications of immunology. He's a member of several scientific societies, and he's part of the editorial board of very prestigious journals. He was a postdoctoral fellow at the, I don't know how to pronounce that, but IRACTA, which is a program aimed to facilitate the progress of postdoctoral candidates toward research and teaching careers in academia. He has undergone extensive training in STEM pedagogy and classroom assessment in a way to improve active learning and professional development of graduate students, postdoc, and early career faculty. Dr. Pullen has an extensive experience in academia and will generously share with us some of his knowledge during his talk, Making Connections, Concept Map for Teaching and Learning Immunology. Nicholas, thank you very much for accepting the invitation to participate in this webinar, and we will be pleased to listen to your talk.
Alrighty, now I think I'm good to go. Sorry, I jumped the gun a bit there.
It's okay.
Thank you again for the invitation. It really is a pleasure. I'd love to be in Argentina as well, but I'm just as happy to be doing this for you now. So thank you for the wonderful introduction. Hopefully you didn't oversell me too much. I did want to start with a little bit more background on where we are. So we're in Greenlee, Colorado, University of Northern Colorado, also known as UNC, about 9,500 kilometers north of you all right now. And we're just about an hour north of Denver, right on the Rockies. So one of the jokes we have here is that we're the UNC Rockies campus because we often get confused with the University of North Carolina. We have no affiliation with them. We also get made fun of because they say we're a plain school, but that's Longs Peak and Mount Meeker from view of, that's Greeley right there. So anyway, I'm in the School of Biological Sciences, which is part of the College of Natural and Health Sciences. And we are one of the three research institutions in the state of Colorado, the other two being Colorado State and the University of Colorado. And we have a slightly different and special mission, and that's grounded in our history as founding as what is called a state normal school, which in the United States back in the day, over a century ago, those were the colleges that trained future elementary, middle, and secondary teachers. So we don't just do that now. We've expanded our mission, obviously, but the point is that informs a lot of our special programs and our research. And the School of Biology has a particularly special program called the PhD in Biological Education. And I mentioned that because that is this program, and I'll say more about a couple of students later, is what has really informed my recent use of the tool that I'm talking about today, which is concept maps. So there's our Western campus, and I'm talking to you right now from about right there. Can you see me? So anyway, it's a pleasure to do this virtually, and back to concept maps. So the objectives that I have for you today are to, first of all, of course, define what concept maps are and what the theoretical underpinning of that is. And then also identify for you or list for you a few things, not comprehensively, but a few things that I use concept maps for that might give you good ideas for, as well as showing you an example activity in a course, as well as seeing examples of the concept maps from that activity, and then also recognize at least one limitation. And I should say that I also want to give you a list of concept map building tools, as well as ways to assess concept maps. An assessment could mean either for grading, or, and I'll show you a case study, for answering or addressing, I should say, educational research questions. And so to start out, what is a concept map? In its basic form, it's what we call a graphic organizer. So what that means is that it gets a student or any person who's drawing it, I draw them as a faculty member to this day, to show the structure, their organization of their knowledge basis, the content knowledge basis that they have. And so I should have backed up and said, you know, my training is in basic sciences, and so I'm a novice when it comes to the cognitive psychology part of this. But my understanding as a novice cognitive psychologist is that it is the organization of one's knowledge, not just whether or not you have a basis of knowledge, but the organization or the structure of that knowledge, that it really allows people to demonstrate that they can accurately use it and apply it. And so more to the theory, Shavelson out of Stanford University published a nice paper a while ago now that basically says that concept maps and cognitive maps are windows into the mind of the person who's generating them. And so basically they're a window into the structure of how students are organizing their declarative knowledge that normally would be assessed by multiple choice tests or fill in the blanks, but the multiple choice tests and the fill in the blanks don't tell you, they tell you whether or not they have an extent of knowledge, whether or not they can recall some specific knowledge, but they don't tell you how they organize it with other things. So this is just a figure from that underpinning where Shavelson is talking about different assessment techniques to assess different areas of knowledge, domains of knowledge that his group defined, where declarative knowledge would be knowing something. Procedural knowledge would be knowing how to use it. Schematic knowledge would be knowing why. And there's actually a fourth domain, which is strategic knowledge, which is the when, where, and how knowledge is applicable. And so he would argue that concept maps, you know, fall into this kind of lower order declarative knowledge. In my experience, now I'm no psychological expert, but in my experience, I would argue that concept maps actually get more into procedural knowledge. So applying relationships and applying new things that a student learns. So back to the definition of a concept map, I sum it up as a concept map is a illustration where ideas or concepts are represented as shapes. And those shapes, you know, could be a circle or a square, star, whatever you want it to be. The shapes or the concepts are then connected by lines, arrows, and so forth that indicate some kind of relationship. And often those lines or arrows will have some kind of process or action verb associated or describing that line or arrow. And so what you end up with is shapes plus connections, so concepts plus their relational actions. Together, these are a proposition. And then multiple propositions interacting with one another on a map usually gets at a larger target idea or topic. So this idea has actually been around for about 50 years now, which still blows my mind that the early 70s were 50 years ago now, by Joseph Novak, who's now an emeritus from Cornell University. And I'll say more about a tool that he spearheaded later for concept mapping. And he would say that concept maps really get students to visually show you meaningful learning or whether or not a meaningful learning intervention has happened. So one of the big questions is what is meaningful learning? And Novak would probably argue that it is the integration of thinking, feeling, acting, so experiences or transformation of your day-to-day life with the specific, you know, quanta of information that you're learning in a class. I think this is just a cute little picture. It's a concept map that's describing what concept maps are. Very meta, if you will. But anyway, that's from King's College London, a blog about concept maps. So go look it up if you like. So if you know a little bit about concept maps, you might already be familiar with, there's a rich literature about all sorts of different kinds of graphical organizers. And one question that my graduate students and I who work with this will often get is, well, how is this different from a spider diagram or a mind map? And so here you see two representations of that. Here you see an actual comparison from visualmapping.com about what are the differences between concept maps and mind maps. Over here on the right, we see an actual depiction. So a mind map is more of a representation, like Okay, so the green circles are concepts, and then you've got your lines to other concepts. It's very what I would call wheelie spokey. Okay, so each individual proposition here is independent of the other propositions. All right, so it tells you something about this central proposition or concept, whatever that might be. Whereas concept maps, you still have that kind of central idea or topic. You've got some spoking outward, but what's really important with someone who's achieving expert level integration about a topic, you start seeing, oh, well, you see new propositions being connected out here among those spokes. And so that's what people will often say separates a mind map from a concept map. I think it's kind of ironic over here on the left that this comparison is more mind mappy than it is concept mappy, if you will. Okay, so what is a concept map? Basics. So there's one that I threw together earlier today, just really quickly. Very simple, a couple of propositions, not a full-on assignment, did it in like 30 seconds. So here we have a central concept, lymphocytes drawn in a box, okay, connected over here to innate lymphoid cells and natural killer cells. Okay, so that's another concept. And then the relationship is that, whoops, that's a little early. The relationship is that these are the lymphocytic cells without antigen receptors. So that together is a proposition. ILCs and NKs are lymphocytic lineage without antigen receptors, whereas B cells and T cells do possess those antigen receptors. So that's a concept map at its basics right there. Now, there's a lot of online tools I'll talk more about. I'll list them off a little bit more later. We're going to get, more aesthetically impressive looking concept maps. So here's the same concept map. I threw it together using a tool called Biorender, which you may or may not be familiar with. We actually have cells now representing instead of those shapes. Okay, so now I have some aesthetic investment in here. I even got B cell receptors and T cell receptors. I was so proud of myself. Anyway, and just to be clear, I know for criticism, I know that's an NK cell with granules and whatnot, but that's the best I could do right now. What I will drive home here is that both of these are equally as valid from an assessment perspective, in my opinion. So I wouldn't necessarily give more points just because one map looks better than the other. They're both relatively accurate in the amount of time given to do it. Okay, so this is my shameless plug just to show my actual bench research. But the real point of this is that maybe we're making concept maps by accident. And I would argue that this idea is really not that foreign to immunologists with all our complicated diagrams that we put, especially in our review papers and then, of course, in textbooks. And over here, you see from a recently published paper from our work where we're basically making a concept map about how the immune system interacts with traumatically injured muscle. Now, what's funny is that we were not intentionally saying, let's go make a concept map of this, but that's what we went to in terms of organizing our knowledge around that topic. Which illustrates kind of like that expert level kind of thinking, which we're always trying to get our students to. And then in another paper over here about, after my own heart, the mast cell, all the different things that we're saying mast cells contribute to. Now, this is more of a mind map because we actually don't have network integration among all of these different topics, even though they could influence one another. So again, the point is that this shouldn't be too foreign and we should start asking our students to start doing it as well. So other than, okay, these are cool pictures, what can you do with them? So really at the apex or at the top of what can you do with them is that you can actually visually see, get a snapshot right away of how a student is organizing their memorized prior knowledge or any new knowledge that they've got. You might have heard of the concept of memory palaces, which are very different, but the idea of like in your mind, imagining placing pieces of information in different parts of a room, for example. Well, instead of keeping that in your mind, you're asking the students to represent that themselves on a piece of paper or on a screen. And so you actually get a snapshot of how they organize their knowledge networks. You can enforce with a snap assignment, you can enforce students to establish new connections that they might not have seen before between concepts that they might have perceived as disparate. It's also a really good formative assessment to catch misconceptions, like if there's a significant error that you would not have otherwise caught because you didn't ask the students about that specifically, or you didn't have a multiple choice question, for example, about that. And then, of course, you can engage in building relationships among new topics, different topics that's kind of addressed already, but also relationships among students if they peer evaluate each other. And then I'll show you a case example where we're using it as a promising research artifact. Now, not research for mast cells or other aspects of immunology, but research on science education. In that case that I'll discuss in a few minutes, you can see students enact systems level thinking, so higher order, the person as a whole or all the physiological systems working together rather than in isolation. And you can, of course, relate it back to either grades or to research, especially, you can measure learning. Of course, what learning is can be quite controversial in some faculty meetings. But Hay, who we base a lot of our research on with concept maps, would argue that learning is change. And concept maps are a great artifact that you can use to document said change. And so from this figure in this paper by Hay, what you can see is they've classified 3 types or 3 categories of learning, or one of them being non-learning, where you take a concept map before you do a teaching event or some other kind of learning intervention, and you say, okay, with all your prior knowledge and instruction of how to draw a concept map, do that. Okay, so we see some concept maps. You do your learning intervention, maybe you teach a new topic, maybe you teach it a different way, maybe you use some different active learning techniques, and you say, okay, now draw another concept map. Non-learning would be no change. You're basically just regurgitating what you already had before, so the knowledge structure remains unchanged. Rote learning, which is a form of learning, there is some change, but it is not moving toward kind of an expert level substantive network change or reorganizing the content. So you see that after the intervention, the student has removed some concepts, has added some new concepts, but really this central concept is still driving the whole map or the whole conception of how this student perceives whatever the topic is, the overall topic is. Meaningful learning, so the transformative learning, relating it to your everyday life, diseases, applications, things that weren't directly talked about in class. Okay, so you have a concept map of basic knowledge beforehand. So for example, definitions about what innate immunity is. You go through some kind of intervention or learning event, and now all of a sudden you're relating disparate concepts accurately, we hope. But this is what we would call meaningful learning because you're reorganizing your knowledge. So again, this is by David Hay, who's at King's College London, if you want to look that up. And so this actually gets at the concept of transformative learning cycle, which I think I've said a couple of times now, where we as the teacher are the alleged experts, right? And so we have these complex, we have a broad base of knowledge, and then we have complex networked ways of integrating that knowledge together. Okay, so we want to impart that upon our students. However, usually science curricula are set up in these very sequential ordered ways that are very linear, and that can lead to rote learning. So you have development of concept maps, but in a very rote kind of linear way. And then, however, what we want to try to get to, and concept maps can help you do this, is get to these students starting to emerge or rearrange their ideas of things in relationships, as relationships are key in new ways, ideally to eventually get them to an expert level of teaching. Ironically, I just caught this morning, if you're interested in learning more about transformative learning cycle, there's a lot more in, outside of concept mapping. There's actually a podcast that was available as of today on insidehighered.com about it. It's pretty cool stuff. So again, the point is that concept maps can be a tool to visualize meaningful change and meaningful learning. And you can actually start to quantify that. And I'll show you ways to do that. So that's all of the theoretical underpinning in a simple example for you. So let's just look briefly at how do we build a map in a particular activity. And so let's imagine, so here in the United States, oftentimes we have 50-minute course periods. You meet Monday, Wednesday, Friday. So that's going to be my basis for this. So what I'd say is, all right, we're in immunology class, pick your favorite topic. You're going to do a concept map for the first time ever. What you want to do, and it's really important, is to introduce the idea of concept mapping to your students And the best way to do this is to give them an example and do it real time with them. I'll never forget when I first learned how to do this. I had no idea what the heck the professor really wanted me to do because they never actually explained what the heck a concept map was. It's literally a drawing of relationships of central ideas and topics and whatever the content is we're talking about. So provide a real-time example. You could give students a basis of terms, or you could just ask them to basically provide all their prior knowledge. But if you do give a list of terms, what I would expect is that the students should contribute more to that, especially if you've already had like a lecture or a class period about whatever the topic is that you're going to concept map on. What I can say is that forthcoming immunology texts will do that. They'll have concept mapping guides and activities, will provide words, but they're not exhaustive lists. So you want to see what the students can do themselves. Some people will give a pre-made map, so like kind of like a fill in the blank map. Some of my colleagues, and I've even done this for lower level, kind of like intro biology courses, just to help students kind of almost like fill out a puzzle. I personally don't like to do that if we're actually going to talk about meaningful change and actually seeing how students organize it, because when you hand out one of those fill in the maps, you're enforcing a certain concept architecture onto the student, and you're not actually allowing them to draw it and construct it themselves. All right, so you introduce it 10 to 15 minutes. Then you could spend about 30 minutes having individual students construct maps. They can do this with pen and paper. They can do it online with a variety of tools. And you could float around either in person in a classroom or through, like Zoom breakout rooms, whatever they call them, and check on their progress and answer questions as you go. And then wrap it up at the very end with end cap questions. Alternatively, instead of doing this at the end, you could break this time up, say at 15 minutes, take a break, let's all get together, talk about what's going on. Do you have any questions? And you could do it that way. With this kind of activity where you're using a whole class period, I would then take those, assess them, and then at a subsequent class period, ideally the next class period, you'd have the students come back and present to some extent their concept maps and their ideas. And you can do peer evaluations this way. You can look for misconceptions, and you can see where your students are. All righty. So say you do get a map, how do you assess it? And so the rest of the talk will focus on essentially 2 case studies of assessment, where you're assessing maps either for grades, which I'll talk mostly about last, or you can actually use these concept maps to do educational research. For example, to see if a certain active learning intervention that you have an idea about works, if you have a hypothesis about that. So first of all, before anything else, make a rubric. Always make a rubric. This will make your life so much easier when you have these, what are inherently subjective things that are being submitted to you. Say, make a rubric. Here's an example rubric from an immune system concept map that I assigned to a general physiology course. And I'll come back to this. So if you're going to collect these for a grade, give the rubric to the students ahead of time so that they know what you want. Probably don't give it to them if you're doing a specific research question that doesn't affect their grades, but you don't want to set the students up for failure. So if it's for a grade for an assignment, I give them the rubric ahead of time. That's mine. It's very qualitative and descriptive. It probably falls into a type of rubric like up here called a traditional concept map rubric from this paper by Watson and colleagues. The title of the paper is Assessing Conceptual Knowledge Using 3 Concept Mapping Scoring Techniques. You can see they're good engineers, and they've started to numericize their measurement of concept maps here to the extent that they even have a new categorical concept map scoring system. I love engineers, but really this actually is an interesting paper if you're interested in going that in depth with quantifying concept maps, just to give you an idea. So always give them a rubric, or always have a rubric at the ready. If you're going to grade, if you're going to do research. Okay, so if you're going to use concept maps for research, that's where I'm going to start. Kind of like a case study of how we've used it for educational research. So I'm one of the instructors for a large enrollment. I consider it very large enrollment, anatomy and physiology course, about 600 students a year, and they meet in large lectures, and then they break up for once a week meetings for about 3 hours in a laboratory setting. And in those laboratory settings, those labs are taught by graduate teaching assistants. So they're teachers, they're just not me or my other professorial colleagues. And there's anywhere from 30 to 40 of those sections running in a given year. And so we were interested in, okay, this course is what is considered colloquially a gateway course, because students get to it and a lot of students don't do well in it. And so we don't want to set students up for failure. We want to do whatever we can to make sure that they are investing into having a meaningful learning experience, which a major facet is getting students to choose to do meaningful learning. And so one way to do this, because this course predominantly serves pre-nursing students, we've developed a problem-based learning intervention. So a lot of medical schools are doing this now and other programs. And the way that we structure it is there's basically a long-running multi-week, you got a patient and they have a lot of problems, and you have to look at all of these problems from the variety of different systems physiologically in this person. And so we instituted this PBL. If you're interested in it, one of them was just accepted with Course Source for publication. The other two are about to go under review. We expect them to be published by the end of the summer. All the teaching materials and everything will be available for it. But anyway, what we were doing with the PBL is we were assessing if it changed a few things. For example, student attitudes, like how their conception of how they felt as a scientist, how they felt as themselves as scientists. Systems thinking skills, so thinking about human physiology not in isolation of individual systems, but a whole that contributes to homeostasis. And then, of course, their basic physiology content knowledge. And the way that we did this research project, it started in 2019. We had 2 comparison cohorts or control groups where we taught the course as it had been historically taught in the lecture in the lab. And then we introduced the PBL treatment into the labs in 2 subsequent semesters that it was offered, and that was our comparison. And so we used concept maps to, we're currently using concept maps, I should say, to assess systems level thinking. We could also use it to go back if we wanted to use it for physiology content knowledge, but we have other measures of that right now. And so this is just a framework of how we're doing this research: content knowledge, systems thinking, and student attitudes. Again, we're using concept maps to address systems level thinking. So the idea is that we're taking the concept maps and we can do 2 things with them. We can look at how concept maps change over a semester in a class. We can also then compare that change over a semester in that class to another class. So for example, from the control to the PBL treatment group. So you might be wondering, how are concept maps used to assess systems thinking? Well, here's an example from the beginning of that class. Probably the first or second week. And this is a very complex, a very good concept map. I've often joked or wondered, maybe the graduate student made it just to fool me. But anyway, so what we're doing is we're actually looking at the complexity of how students arrange ideas about a given overall topic, and we're quantifying that. So the way that we do that is we quantify the shape of the map, the number of concepts, so the number of boxes or circles or whatever that are your keywords, and then the number of written descriptors of the relationship lines. And actually, we quantified the total number of relationship lines as well as those that had descriptors. So that's for our purposes, for research purposes, and we can actually quantify that and watch it change with treatment. Excuse me. The students, it's also useful for the students because it's a formative assessment guide for them. So the students should receive feedback from their teachers, the teaching assistants, and then that provides an opportunity for the student to then apply that feedback to future concept maps. So it's important always to do pre-post kind of measurements as well around an intervention. So the idea is that with the feedback, the students can learn how to improve and develop their systems thinking strategies. So this is where I just want to make clear that concept maps on themselves have been very, very heavily studied and themselves are an active learning intervention that can be used. What we're doing here is we are then using those concept maps and seeing if we see changes in the learning or the representation of those maps with another intervention, problem-based learning, on top of that. Get a drink of water. So then we might wonder, okay, so how do you actually quantify this and deal with the map complexity? So this is how we do it. We actually use part of that Hay paper for our inspiration. Here you see a figure from that paper representing several facets of the maps. And what we did is we developed a complexity score that took into account shape. So over here, if we look at the Hay example, whether or not it's very spokey looking, very chain-like, or if it has that kind of expert ideal network kind of connectivity in it, we assigned arbitrary scores to that. And so a chain would be a level 1, a spoke would be a level 2, a network would be a level 3. Now, just to be clear, there actually is some debate on whether or not chains or spokes are more complicated. We made the call that the spokes are more complicated, or more complex, I should say. But the argument is that spokes are actually relatively simple, for what I said about mind maps earlier. Each of these propositions are technically independent of one another. Whereas in a chain, if we look at whatever this concept is down here, this total proposition is completely dependent on all those before it. So that's actually pretty complex organization of knowledge. It's a very linear rote organization of knowledge, but there is an argument that it could be more complex. We also looked at what we call the hierarchy. And so we gave a score to the longest chain. So the longest chain, uninterrupted chain, we gave a score to that. And then we also counted up the total connections and the connections that were labeled. We attempted to throw some other subjective values in there, like the actual subjective values. So I'd say these are mostly objective. We tried to throw subjective values in there, like the actual grades on lab reports and quiz assignments, that point in time grades, as well as the validity of concept maps. But we actually have been really struggling with this, especially validity. So we've actually removed these from our complexity scores right now. And the reason for that is because we're seeing a lot of variability in the feedback of the teachers, the feedback that the teachers provide to their students. And so that's one of the main limitations of concept maps, especially with large enrollment. They can get so big and there's so many of them that if you don't have all of your teachers who are participating in it and instructing it on board, you can get widely variable feedback levels, from no feedback to, as you see down here, simple error correction, very rich feedback to promote student thinking, or kind of platitudinous praise. And so the idea here is, or the problem idea here is, that if students don't get the same or equivalent feedback, equitable feedback across sections, it makes it really difficult just to use the concept maps. But we haven't given up on that yet, because as of a few days ago, actually, my grad students leading this project have found that even just getting feedback, even if it's not necessarily good feedback, that actually correlated with a substantial change and substantial positive, more networky change in concept maps. Okay, so the point is, it's really important to get everybody on board with what concept maps are if you have a big audience or diverse group of people. We've been able to do this very easily or very well with our raters. So we actually hire students and train them to actually quantify these maps for us, and the inter-rater reliability is pretty high. The issue is the feedback that the instructors are giving to the students. And we've tried some professional development to mitigate that to some extent. That's a whole other topic, and there's a very rich literature on effective professional development, but that's separate. Okay, so we're in the land of COVID. How can I make concept maps if I don't want to do pencil and paper? There's plenty of stuff online. This is not an exhaustive list. CMAP is the one that Joseph Novak himself endorses and runs. Biorender is the thing illustrated over here that I showed you. This has a subscription kind of architecture to it, but it's not required. So students can go in and do, for private use, make concept maps, for an assignment with a free account. They don't have to pay for that. I always recommend draw.io. This is totally free and it plugs into Google Drive. In the NovaMind is one I don't have experience with, other than sometimes students give it to me or submit assignments having used it. This one does cost money, and for whatever reason, I don't know, I guess other faculty have used this at some point. Some students will already have subscriptions to that. So let's take a look at the very end of this at some example concept maps from a very recent assignment in immunology. So you actually see an example concept map in the background from a student who had never taken immunology until they got to this immunology module in a physiology course. And that was drawn using the draw.io platform that I just mentioned. And so going back, here was the rubric that I provided the students ahead of time for this. And then on the left are their instructions. And so you see, I didn't give them a bank of words. I actually treated this as a homework assignment. I didn't do it in class. So you're seeing a second way to do this activity. And so I gave them 4 days to do it. And it was right before the final exam as we were finishing the immunology quarter of this class. And you see that I did give some guidance. I wanted some innate immunity, some adaptive immunity, immunological overlap and disease. That could actually be 2 separate topics. And then I put a quality thing in here. I never give anyone 0 points. This is just to make them, kind of enforce kind of taking it seriously. And so here are the instructions. This is an example or a test case. I did not actually train the students ahead of time to do it. All I did was give them a link to the Wikipedia article on concept maps, which, by the way, is what Cornell University cites in their blog post about Novak. And so here's some examples. Here's one using draw.io. You see it's quite, you might see, or I'm going to tell you, this is what we would call quite spoke-like or spokey. There's not much network integration. You can see a little bit up here, okay? And there's some off the screen, unfortunately. But for the most part, you have these families of content that are branching out and relatively independent. Here we see one that was made, I think it was actually made with Biorender. What we see is more of again, a spokey kind of mind map where you have different families of concepts radiating out from this central concept, but really no feedback among the different branches. You do have really great feedback over here. They were really into whatever this was over there, inflammation, innate immunity, and then a little bit over here with adaptive immunity. Can you tell that I am an innate immune kind of person? And so that's probably where my bias is in teaching immunology. But anyway, there's an example of a very kind of spokey, more of a mind map kind of thing. So this kind of map actually causes quite a bit of controversy and requires variability scoring when we're like doing this for research, which again, this was just an assignment, because I would say this is not as complex as it could be. It looks complex, but you don't have a lot of networking there. This is very similar to that prior slide, except this one is just words, which is totally fine. I would not take points off for not drawing boxes. That's okay. I don't know what this one was made in, but what we see here is we see more networking, especially with how inflammation is related to these topics over here. Antigen presentation, obviously. One of the issues with this map, however, is that the student is relying heavily on essentially paragraphs of information when the idea is to kind of sum this up into individual kind of words. Here's a really beautiful example from Biorender, but you see that it's really just a strictly hierarchical, it's not a kind of mind map. They really, really were jazzed about the receptors and the cell types, which is great. But what could be worked on here is integration of how these cells would influence other cells in this hierarchy. Now, what is also great here is that you see a lot of descriptions on these linkers, which you didn't see on some of those before. This is getting toward a level 3 kind of map. You see descriptions on the linkers. You see a lot of networking across disparate topics. This one I just showed has a lot of, you might not be able to see it here, but it has a lot of that kind of networking. And we see that this was done with pastels on a canvas, actually. And then this, I would say, was probably the exemplar from this particular class. Aesthetically, it looks very different from the Biorender one, but what you can see is this person probably has a lot of prior knowledge of immunology before they got to this class, and you see an expert level kind of integration of knowledge here. So just to close this out, some quick notes about this particular assignment. As homework, you can see that the effort level was pretty high, and I would say it was pretty much 90% across the board, high level of effort. For those were just examples. And the reason I mention this is because I've been getting pushback from colleagues recently saying, oh, my students would never make something like that. I would argue, at least in my experience, that's nonsense. The students did meet the rubric's descriptive requirements, so pretty much everybody got a good grade. However, because I didn't give instructions, a lot of people, I'd say about half the class, made more kind of like spider mind map, kind of spider diagrams, and didn't do a lot of networking because I wasn't necessarily assessing networking. And then finally, the most aesthetically impressive maps are not necessarily the most valid. So I'll be quiet there. I'll wrap it up there. I'm not quite yet. I have to acknowledge a couple of graduate students, Alex Vita and Emmy Royce, who've been running the PBL project. They are major influences on my use of concept maps. Alex is actually, they're both PhD candidates about to finish. Alex is actually has a major bench component in my lab. She's studying rheumatoid arthritis, but also does bioed research on the side like I do. Emmy is an example in our PhD program that I mentioned is pretty unique, where she focuses exclusively on the social scientific aspect of STEM or science teaching. And so finally, I'll acknowledge that the PBL work is supported by UNC assessment grants. There's my contact information. And I'll just say at the end that I can at least speak for myself that at least at this point in time, my novice knowledge of concept maps and the cognitive psychology behind that, I learn the most. I get the most meaningful learning from my students. So once again, thank you for the opportunity. I'm open for any questions you might have.
Thank you, Nick. Thank you, Nicholas. Can you hear me?
I can hear you.
Yeah. It seems that there's a lot of questions, but there are questions that pretty much is like it seems at the beginning very difficult, you know, to incorporate this concept of the concept map. I mean, I think we usually use it spontaneously when you write the review, for example. You have a tendency to connect concepts when you want to write a review, not only a teaching point, I think. This is novel for us to apply this kind of strategy for teaching. So we have a lot of questions. Basically, you can read here. Can you read also the chat?
Yeah, I'm looking at them right now.
Our experience at first.
Okay, so
No,
there are
here. Sorry, here. A little bit lower, yes.
Okay. Oh, I'm sorry. Am I still sharing screen?
It seems that building concept map is a good tool to evaluate or assess the knowledge of the student, but when a new topic is developed, do you show them how to build a concept map?
Yeah,
Yes.
so there's a little bit, you know, I breezed through that. So there is a little bit more to concept mapping, especially in the beginning, if the students have never seen it before. And really, it's really important to just draw one out for them, probably more complex than what I showed. You just spend 5 minutes drawing one out, and I don't explicitly say, oh, and you know, this is a really good concept map because it loops back. I lay the groundwork by doing it, but not being explicit about it. So, for example, integration of topics, you know, integration of myeloid cells involved in antigen presentation with how that might work with different kinds of lymphocytes. I'll draw lymphocytes and myeloid cells as disparate, you know, areas, but then I'll start linking them and networking them together. And so that kind of like lays the groundwork for doing that. And what we see is that with our research project, which hopefully it's not published yet, hopefully will be published soon, we do see that even without that kind of intentional, like you need to draw networks, the students get there eventually. Now, another big part of this is if you go into the literature about what meaningful learning is, a major facet of that is that students have to choose to do meaningful learning. They have to be invested in the process. And what we see is that you start seeing that networking when they get invested. So that's why we're using concept mapping to measure whether or not problem-based learning is effective, because the idea behind the problem-based learning is that it gets the students more invested. They're all pre-nursing students. They don't care about, you know, Apposed syndrome that some immunologist is studying, because they may never specifically see that. What they care about is seeing a patient who's undergoing broad spectrum kind of disorder. Then we use the concept map as a tool to see if they're actually being invested in that learning process. And you start, the hypothesis is you start to see that networking when they get invested. They choose to participate in that meaningful learning. Right. So that's the whole point I was trying to drive home with that. I mean, you could totally, if you want to, say networking, that's what we want to see. That in of itself could be a learning prospect, but that's not how we were using it. I'm not saying that's wrong. That's just not how we were using it.
Right.
Good question.
So another question is, do they build math for each subject, or this tool is more useful for some topic than others, for example? And especially the question that I also share with this participating in the talk is, how do you just evaluate? How do you just, I think it's just kind, for me, it's like subject, I mean, it's like not very objective. You see who students develop the best concept map. I mean, I think it pretty much is because I think the student could do that better than myself. I mean, how do you evaluate that?
So I'll hit the first question first, which was, can you use this for every topic, or is it better suited for certain topics? You can use it for as many topics as you want. In the AMP course that I mentioned, we use it for every physiological system. In my general physiology course, I only use it for immunology, because this is, in my opinion, immunology is the toughest topic that they face. And so the feedback I've gotten is that students really value being forced to make those relationships with that activity. So it's really up to you whether or not you want to use what you want to use it for. I've used it across the board. Now, to the question of how do you evaluate this if it's for a grade? Yes. You have hit probably, I don't think I made it clear. That's the second big limitation of these, is that you can really get lost in the weeds pretty quickly if you don't have a good rubric that you are going to stick to, that you've developed. And so that's kind of what I was getting at with the very end, where I said, in general, students, just a quick look, because I provided the rubric ahead of time, they all got good grades because they met the demands of the qualitative demands of the rubric. And so that's what I wanted to get out of it for that particular assignment. It was really for them to study for the final exam. It wasn't, and for me to look at and say, oh, wow, they've got, dendritic cell expressing full-on T-cell receptor complex. If I see that, whoa, we got to stop. I don't know if you saw the instructions that said, I basically look for stuff like that. We got to stop and we got to talk. There's a big problem here.
Yeah. So if you start with a rubric, right? This is the beginning. And then you just check how close or how the interpretation of this rubric is better for each student, right? So when you mean rubric, you mean like it's instructions? So where they have to start to develop the topic, I mean?
Yeah, I'm sharing my screen again. Let me just go back to it. So there's the actual rubric.
Right.
Okay. And so, it has embedded instructions.
Right.
And then over here, I have kind of more general instructions. So the way that this appears in the learning management system is the more general instructions are on top, then there's a rubric that gets more resolved or granular. And you see that I assign points based on this. And so what would I consider a major concept under innate immunity? Well, if they're missing like the role of macrophages, neutrophils, or dendritic cells, they're going to get a point off for that. If they're missing NK cells and viruses are involved in the map, they're going to get a point off for that.
Right.
So it is absolutely subjective. And this gets to the point that I made about TA variability with our research project.
Right.
And so if you have a lot of people doing this, you want to make sure that everybody who's teaching it is on the same page.
Yeah.
If you're the only person doing it, you want to make sure you want to be upfront and clear about your expectations with the students when you give them the assignment.
Exactly.
As well. Yeah.
I think it's more than a strict way to evaluate. It's better to, it's useful as a tool to understand how deep the knowledge from each student is.
Yeah.
I mean, in my opinion, right? Yeah,
I didn't show it, but this is how we can more specifically quantify a map. And so this was using that, this was for our research project using that scale. So this particular map is quantified here. Takes more time. So if you got the time, you can go as deep as you want with this. That's the other point, is that this is a very rich research artifact on top of being, hey, show me your work. Are you on the right path kind of thing?
Right.
Yeah.
If they do understand better, they're gonna develop a concept map more complex and more high quality, right?
Yep. Yeah. That's the idea.
Okay.
Ceci, I have a question, Nicholas. It's a comment actually. Because of course the student at the beginning probably they can make or just a very simple type of connection maps or connected map, but at the end of the course, I think that will be a type of evaluation that you can do as the global or whole concept of the subject. You can maybe integrate all the course, and at that point, I think could be a good strategy to make an evaluation with the concept map. I think it could be very, very useful in that way.
Exactly.
I have colleagues who have argued that it's a better summative assessment if you use it that way. So you use a beginning to the end. It's a better summative assessment, according to some, than giving a classical exam, if you will, because you're actually seeing them demonstrate improvement in their connections. And so that's up to the individual instructors for how to evaluate it. I totally agree with you on that. And that's exactly what we're doing. So with that research project, that's exactly what we're doing with the grades in the labs. That's a contribution to their grade, because they start at the beginning of the semester and go through the whole semester with this PBL project, this one patient, and they develop maps, and the maps get more complex. And that contributes to their grade. It's not the only part of the grade, but it contributes to it for sure. Yeah.
Okay. That's great. We are all learning from you.
Yeah, it was very,
like I said, these are not my ideas. These are all based. These are all based in Joseph Novak and Hay and the references that I gave you there. Yeah.
Yeah, but for us, it's just you. We're just getting that from you. Thank you, Nicholas. That was very interesting.
Thank you.
Thank you very much. Yeah.
Yes, for us, we are not very accustomed in the Argentinian Society of Immunology of having this type of webinars. We usually have webinars related to fundamental immunology or clinical immunology, and these are the first time that we are doing this type of webinars focusing in how to teach or new tools, or trying to share experiences of how to teach immunology. So for us, it's really a new experience. Most of us are also teachers in many universities, so it's very nice to hear how you use different tools in other parts of the world. So thank you so much,
thank you again for the invitation. I apologize for plugging the basic immunology in there. I had to put it in there.
No, Nicholas, our university is also UNC.
Oh, it is?
Yes, it's Universidad
How many
Nacional. It's
I can't tell you how many phone calls and emails I get about
but different meaning.
Located.
It's Universidad Nacional de Córdoba. So UNC like you guys.
Yeah, we're the better UNC, right?
Yeah, we are the better in the world. Thank you, Nicholas.
It was a pleasure. Thank you so much.
Thank you, Nick. Thank you, Cecilia and Antonio, and all the people who have participated through the YouTube channel. Thank you so much.
Thank you.
Bye-bye.
Thank you. Bye-bye.
Gracias, chicos. Excelente
Bueno.
todo, ¿eh? Muy bien, gracias.
Buenísimo. ¿Ya se cortó la grabación? Sí.